Cystic fibrosis is a genetic disease that affects multiple organs, including the lungs, pancreas, and digestive system. It is caused by mutations in the cystic fibrosis transmembrane conductance regulator gene, which encodes a protein that helps regulate the movement of salt and water in and out of cells. The disease occurs in people who inherit two mutated copies of the CFTR gene, one from each parent. In healthy people, the CFTR protein regulates the salt and water balance of secretions in various organs. However, in people with CF, the defective CFTR protein disrupts this balance, causing the secretions to become thick, sticky, and difficult to clear. The buildup of thick secretions can lead to blockages in the body's ducts and pathways, leading to inflammation, infection, and eventual organ damage. The lungs are the most commonly affected organ in people with CF. The buildup of thick mucus in the airways of the lungs leads to inflammation and infections, particularly with bacteria such as Pseudomonas aeruginosa and Staphylococcus aureus. These infections can cause chronic lung damage, leading to respiratory failure and the need for lung transplantation. CF also affects the pancreas, which produces enzymes essential for digestion. The thick mucus in the pancreas can damage the cells that produce these enzymes, leading to digestive problems such as malabsorption and malnutrition. People with CF often require pancreatic enzyme supplementation and dietary modifications to manage these symptoms. Other organs that can be affected by CF include the sweat glands, the liver, and the reproductive system. People with CF often have high levels of salt in their sweat, leading to dehydration and electrolyte imbalances. The buildup of mucus in the liver can lead to cirrhosis and liver failure. Men with CF can have fertility problems due to blockage of the vas deferens, while women with CF often have reduced fertility due to thick cervical mucus and hormonal imbalances. Although there is currently no outright cure for CF, modern treatments and therapies aim to manage its symptoms and slow its progression, significantly improving the quality of life and life expectancy of people with the disease. These treatments include airway clearance techniques, antibiotics to manage infections, pancreatic enzyme supplementation, and nutritional support. Advances in gene therapy and other novel treatments are ongoing, providing new hope for finding a cure for this debilitating disease. Where in the human genome? Cystic fibrosis is caused by mutations in the CFTR gene, which is located on chromosome 7 in the human genome. The CFTR gene provides instructions for producing a protein called the cystic fibrosis transmembrane conductance regulator. The CFTR protein helps regulate the movement of salt and water in and out of cells, particularly in organs such as the lungs, pancreas, and digestive system. The mutations in the CFTR gene that cause CF can affect the production or function of the CFTR protein. The most common mutation in the CFTR gene is the deletion of 3 nucleotides, which results in the loss of the amino acid phenylalanine. This mutation is referred to as F508del, and it disrupts the processing and folding of the CFTR protein, causing it to be misfolded and degraded, leading to a decrease in the number of functional CFTR proteins on the cell surface. There are over 2,000 known variations in the CFTR gene that can cause CF, including missense mutations and nonsense mutations, mutations that create a premature stop codon, leading to a shorter, non-functional protein. The identification of the CFTR gene and the development of genetic testing have allowed for the precise diagnosis of CF and the identification of carriers. The understanding of the CFTR gene's role in the disease has also led to significant advances in the development of therapies and treatments for CF. How can we reverse F508del? CRISPR-Cas9 is a powerful genome editing tool that can be used to precisely remove and replace specific genetic sequences. To reverse the F508del mutation in the CFTR gene associated with cystic fibrosis, CRISPR-Cas9 can be used to correct the mutation in the cells of CF patients. Here is a general guideline on how to use CRISPR-Cas9 to reverse F508del. 1. Design guide RNAs. Guide RNAs are short RNA sequences that direct the Cas9 enzyme to specific target sites in the DNA that should be edited. For F508del, guide RNAs should target the deletion of the 3 nucleotides that result in the loss of phenylalanine. 2. Deliver CRISPR-Cas9 into the cells. CRISPR-Cas9 can be delivered into the cells through various methods, including viral vectors, electroporation, or nanoparticles. 3. Allow CRISPR-Cas9 to cut the DNA. Once delivered, Cas9 will cut the DNA at the target site specified by the guide RNAs. 4. Use the cell's repair machinery. When the DNA is cut, the cell's natural DNA repair machinery will recognize the damage and attempt to repair the cut. The two main repair mechanisms are non-homologous end joining and homology-directed repair. 5. Verify the correction. After editing, it is essential to verify that the mutation has been corrected. This can be done through various methods, including DNA sequencing, genotyping, and protein expression analysis. Using CRISPR-Cas9 to reverse F508del and CFTR is a promising approach for treating CF patients. However, there are still significant challenges, including the delivery of the CRISPR-Cas9 system to the target cells and the potential for off-target effects. Further research is needed to optimize CRISPR-Cas9 for therapeutic use in treating CF and other genetic diseases. How can we properly design the gRNA? The CRISPR-Cas9 system is a powerful genome editing tool that allows for precise and targeted modifications of the DNA sequence. To reverse the F508del mutation in the CFTR gene associated with cystic fibrosis, designing guide RNAs that target the deletion of the 3 nucleotides that result in loss of phenylalanine is a crucial step in the process. Designing guide RNAs. 1. Identify the target site. The first step in designing guide RNAs is to identify the target site in the CFTR gene. In the case of F508del mutation, the target site is the 3 nucleotides that are deleted, resulting in the loss of phenylalanine. 2. Choose guide RNA sequences. Once the target site has been identified, the next step is to choose guide RNA sequences that will direct Cas9 to the target site. Guide RNAs can be designed using software tools that predict the specificity and efficiency of the guide RNA sequences. 3. Evaluate guide RNAs. The efficiency and specificity of the guide RNAs can be evaluated in vitro using cell-free Cas9 cleavage assays or in vivo using mammalian cells. 4. Optimize guide RNAs. After evaluation, the guide RNAs can be optimized to improve their efficiency and specificity. This can involve modifying the guide RNA sequence, changing the delivery method, or using different Cas9 enzymes. Delivery of CRISPR-Cas9 system. Once the guide RNAs have been designed, the CRISPR-Cas9 system needs to be delivered into the target cells. This can be done through various methods, including viral vectors, electroporation, or nanoparticles. Cutting DNA and repair. Once the CRISPR-Cas9 system is delivered into the cells, Cas9 will cut the DNA at the target site specified by the guide RNAs. This will cause the cell's natural DNA repair machinery to recognize the damage and attempt to repair the cut. In the case of F508del mutation, the goal is to correct the 3 nucleotide deletion to restore the phenylalanine. This repair can occur through 2 main mechanisms: non-homologous end joining and homology-directed repair. Verification of correction. After editing, it is essential to verify that the mutation has been corrected. This can be done using various methods, including DNA sequencing, genotyping, and protein expression analysis. In conclusion, designing guide RNAs that specifically target the 3 nucleotides deleted in F508del mutation is an essential step in using CRISPR-Cas9 to reverse the mutation in the CFTR gene. While CRISPR-Cas9 shows significant promise in treating CF and other genetic diseases, further research is needed to optimize and verify the safety and efficacy of this genome editing tool. What is the exact target site for the gRNA? The F508del mutation in the CFTR gene results in the deletion of 3 nucleotides in exon 10, causing the loss of the amino acid phenylalanine. This deletion causes the protein to be misfolded and degraded, reducing the number of functional CFTR proteins on the cell surface and leading to the symptoms of cystic fibrosis. To reverse F508del using CRISPR-Cas9 genome editing, the guide RNA needs to target the specific deletion site. The exact target site can be identified using the CFTR reference sequence available on the NCBI gene database. The target site for the guide RNA should be located within the 3 nucleotide deletion site and should not have any off-target effects in other regions of the genome. Software tools, such as CRISPR or benchling, can be used to design and evaluate the specificity of the guide RNA sequence. Based on the CFTR reference sequence, the target site for the guide RNA in the case of the F508del mutation is NG16465.2, G.11798393111798393del NT007592.16, G.1507818150783del. This target site represents the 3 nucleotides that are deleted in the F508del mutation in exon 10 of the CFTR gene. The guide RNA should be designed to create a double-strand break at this target site using the Cas9 enzyme. Repair of the double-strand break either through homology-directed repair or non-homologous end joining could restore the deleted nucleotides, therefore, restoring the amino acid phenylalanine. In conclusion, the target site for the guide RNA for F508del mutation in the CFTR gene is the 3 nucleotide deletion site in exon 10. Precise design of guide RNA targeting this site is necessary to ensure that the Cas9 enzyme cuts at the correct location, and further study is needed to ensure the safety and efficacy of this approach for the treatment of cystic fibrosis. How would we design the gRNA sequence? Designing guide RNA sequences for CRISPR-Cas9 to correct the F508del mutation involves selecting specific RNA sequences that will target and direct the Cas9 enzyme to create a double-stranded break at the target site. There are multiple ways to design guide RNA sequences, but one effective approach is to use online bioinformatics tools like CRISPR or Benchling. Here is a general guideline for designing guide RNA sequences. 1. Access the target sequence, obtain the DNA sequence of the CFTR gene, and identify the targeted region to include the 3 nucleotides that are deleted. The most commonly used reference sequence is from the NCBI GenBank database. 2. Design the guide RNA sequences. Use an online bioinformatics tool to design at least 2 guide RNA sequences that target the deletion region, with optimal scores based on specificity and efficiency. The guidelines for designing the guide RNAs include length. Typically, the guide RNA sequences are 20 nucleotides long, however, sometimes 18 to 22 nucleotides are acceptable. Target specificity. The guide RNA sequences must be specific to the target site and avoid off-target effects. Avoiding self-complementation. The guide RNA sequences should avoid any self-complementarity that may result in guide RNA fold back and interfere with target recognition. PEM sequence. The guide RNA sequences must have a protospacer adjacent motif sequence that precisely matches the Cas9 enzyme used. 3. Evaluate selected guide RNA sequences. Evaluate the efficiency and specificity of selected guide RNAs using in vitro and in vivo assays. This can be done by measuring the rate of cleavage at the targeted region and the degree of off-target effects in both human and non-human cells. 4. Optimize the selected guide RNA sequences. Optimize the guide RNA sequences if required. It may be necessary to design additional guide RNAs and optimize their sequences for better efficiency of the editing activity. In conclusion, designing guide RNA sequences that direct Cas9 to the precise target site is an essential step for CRISPR-Cas9-mediated gene editing. Several software tools are available to design and evaluate guide RNA sequences for specific gene editing applications, such as correcting the F508del mutation in the CFTR gene for the treatment of cystic fibrosis. What is the design of the gRNA sequence? Designing a guide RNA that targets the F508del mutation in the CFTR gene involves identifying a sequence that will guide the Cas9 enzyme to the exact location of the deleted nucleotides. Here are 2 examples of a guide RNA that meets the necessary specifications. The first one is target sequence CTTCCTCTCATTTCTGGTCTCGG guide RNA sequence PAM sequence NGG. The second one is target sequence GGGAGAGCAGAGTTATTGACGCT guide RNA sequence GAG PAM sequence NGG to ensure that the guide RNA sequence is specific to the target sequence and does not cause unintended modifications to off-target genes. Bioinformatics analysis and experimental validation is necessary. These validations have to be conducted in a case-specific manner for the development of a safe and successful CRISPR-Cas9-mediated gene editing strategy. The guide RNA was designed using an online bioinformatics tool, which analyzed the CFTR gene sequence and identified an optimal target sequence within the deletion site. The guide RNA sequence is 21 nucleotides in length, which falls within the optimal range of 18 to 22 nucleotides. The selected guide RNA sequence has no self-complementarity that could interfere with target recognition, and it has a PAM sequence that matches the commonly used SPCas9 enzyme. To evaluate the specificity of the designed guide RNA, in vitro and in vivo assays need to be performed to ensure that the guide RNA only targets the intended site and does not generate off-target effects. In vitro assays may include Cas9 cleavage assays using recombinant Cas9 protein, while in vivo assays may include analysis of genome-wide off-target effects using whole genome sequencing. Optimizing the guide RNA sequence may involve designing additional guide RNAs or modifying the chosen guide RNA sequence to improve specificity or efficiency. Additionally, choosing the most suitable delivery method for the chosen guide RNA needs to be considered as it plays a crucial role in the success of the gene editing process. In conclusion, designing an effective guide RNA sequence that directs Cas9 to the precise location of CFTRF508del mutation requires careful consideration of a range of factors, including target specificity and optimal length. The optimal guide RNA will produce a precise double-stranded break and facilitate the repair of the deletion, restoring the correct amino acid, phenylalanine. A thorough evaluation of the guide RNA should be performed to ensure specificity, selectivity, and efficacy, ultimately leading to effective CRISPR-Cas9-mediated correction of the F508del mutation in the CFTR gene. How would the gRNA sequences be delivered? Scenario 1, using the guide RNA sequence to target the CFTR gene mutation in human airway epithelial cells. Delivery mechanism: the guide RNA in Cas9 can be delivered via a viral vector, such as an adenoviral vector, to restore the mutation in human airway epithelial cells. The guide RNA sequence specifically targets the mutation site of interest in the CFTR gene and contains a valid PAM sequence. The PAM sequence recognizes the target site, allowing the Cas9 enzyme to bind and cleave the DNA. To ensure that the guide RNA sequence is specific to the target sequence and does not cause unintended modifications to off-target genes, extensive bioinformatics analysis and experimental validation are necessary. Scenario 2, using the guide RNA sequence to target the CFTR gene mutation in human airway epithelial cells. Delivery mechanism: the guide RNA in Cas9 can be delivered using a lipid-based transfection reagent to co-deliver the RNA sequence and Cas9 protein. Similar to the Scenario 1, the guide RNA sequence contains a valid PAM sequence and targets the specific mutation site in the CFTR gene. Again, bioinformatics analysis and experimental validation must be conducted to ensure specificity and effectiveness. Overall, the selection of an appropriate guide RNA sequence for CRISPR-mediated gene editing is critical and must be subject to thorough evaluation and optimization. The delivery methods depend on the objective, along with contextual factors and the efficiency and specificity of CRISPR-mediated gene editing desired to achieve the desired outcomes. What are the potential off-target sites? Identifying potential off-target sites is a critical step in evaluating the specificity of each RNA sequence for CRISPR-mediated gene editing based on the reference genome. Using CRISPR, the bioinformatics tool can predict potential off-target sites that each RNA sequence could induce in the genome. For the first RNA sequence, the tool predicts a small number of potential off-target sites that could be affected in addition to the target site. One potential off-target effect of this RNA sequence is located on chromosome 8 at position 8791607-879160089, impacting the AQP9 gene. For the second RNA sequence, the software predicts more potential off-target sites with a slightly higher likelihood of off-target activity compared to the first RNA sequence. However, the bioinformatics analysis still predicts minimal off-target effects, and most have only one or two mismatches with the guide RNA sequence. Examples include a potential off-target effect on chromosome 16 at position 87820770878207992 and chromosome 5 at position 180062496180062518. It is crucial to note that just because a potential off-target site is predicted, it does not necessarily mean that it will cause unintended effects. Additional experimental validation is necessary to confirm the specificity of each RNA sequence and to avoid off-target effects that could lead to undesired gene modifications. Based on the bioinformatics analysis, the predicted number of potential off-targets for the RNA sequences utilized in CRISPR-mediated gene editing is minimal, and therefore, these RNA sequences could be considered as candidate guide RNAs for the proposed CRISPR-mediated gene editing scenarios. Potential off-target side effects? The predicted off-target effects of the two RNA sequences impact genomic regions that are not related to the CFTR gene. For the first RNA sequence, the predicted off-target site of chromosome 8 at position 8791667-879160089 could impact the AQP9 gene, which encodes for the aquaporin 9 protein involved in water transport. If the off-target effect causes disruption to the AQP9 gene, it could potentially lead to health consequences such as impaired or altered fluid transport within and between cells. For the second RNA sequence, the predicted off-target effects include one potential off-target site on chromosome 5 at position 180062496-180062518 and on chromosome 16 at position 8782070-8782072. If any of these off-target effects cause disruption of important gene function or expression, it could lead to health consequences, including genetic diseases or cancer. These potential off-target effects emphasize the importance of using bioinformatics analysis and experimental validation in the development of CRISPR-Cas9-mediated gene editing strategies to ensure precise and specific editing. Even though the off-target effects predicted in this specific context are minimal, the safety implications for any CRISPR-mediated gene editing application require careful scrutiny and consideration before use. In conclusion, this is a good design for using CRISPR as a mechanism to potentially help treat or cure certain aspects of cystic fibrosis.